Hello everyone, my name is Olivier Clerc, I am from Switzerland. I am very happy to be with you here this year for this TA workshop. I wish you a wonderful year. It will certainly be a better year than last year. And I would like to talk to you about meta-analysis. This is a topic that I really like because I had the opportunity to do a few meta-analysis and I could also take a course about it. So I'm super enthusiastic about it. And I would like to show you a little bit what it is and how to do it, just a little overview. And maybe after this, you'll want to do a meta-analysis yourself. Let's have a look at it. First, what is that? Actually, when you do a meta-analysis, you want to summarize current evidence to have a final conclusion about what already exists. But actually, a full meta-analysis has two parts. There is a systematic review and then the meta-analysis itself. What is that? Well, the systematic review is a review of all current evidence. You are not going to search for papers and information at random. You want to be systematic, to search the literature completely, to find all relevant articles, to exclude irrelevant articles, to really get the correct ones. And if possible, you want to extract numeric values out of these articles to do a numerical analysis for a quantitative summary, which is the meta-analysis part of it. So again, this is no cherry picking. This is really a planned approach. You need a strategy on how you are going to find those studies, how you are going to search the literature, how you are going to summarize this data with numbers, and how you are going to analyze it. And also there is a quality assessment, which is an important part of it. But let's have a look at more details on it. First, why? Why would you like to do a meta-analysis? Well, again, this is when you want to summarize current evidence because there are many studies and maybe some show that the new treatment is not effective, the other one show that this is effective, but you don't really know, this is inconclusive, or maybe the results are not significant, so you need more power, you want to gather many studies together to show what they can say all together and to find the global conclusion to everything that exists until now. This is also very important to keep other researchers up to date because there are so many publications now. I mean, you cannot read everything, it's not possible. So we are very happy if someone makes a summary for you. And also you can dig into the data and have a look at what are the problems. Maybe the differences between studies are explained by differences in methods, or maybe different patients, different approaches, different analysis. So you can have a look at this and to understand what do we have, what do we need, how should we focus the next research? Maybe we should focus research on these specific patients or focus on that approach because data until now are a bit vague about it, so we need more. This is all aspects that you can see. So you have many things that you can analyze and have a look at in the meta-analysis. Actually, it's very interesting. But first and foremost, you need an excellent idea. You need a topic. You need to think about what you are going to do. You need a topic for which there are already information, data, so something to gather, but maybe not too much, for which there is no evidence until now. So again, the data are conflicting, are not significant, or maybe only some patients were explored and not everyone. So you want to add something new to what is known using gathering of previous data. And sometimes there is already a meta-analysis, but it may be worth doing it again, completing it, because maybe there are new exciting results. Then you think, well, it will change everything. So let's take the new studies and update the meta-analysis. But of course, it's important to have a look at what is already known. If there are already 3 recent meta-analyses on this topic, so maybe you would want to do something else. So that's where you should have a specialist with you to give you advice on what is a currently hot topic for which there is still room for meta-analysis. That's quite an important part of it, finding the brilliant idea that will trigger the meta-analysis. And then, how do you do that? There are different ways of describing it, but I like this one. This is the one with 9 steps. So first, again, you need to find a topic, and then you need to make a plan. You have to plan the review protocol. You have to think about what is known, what you should know, how we are going to find studies, how we are going to extract the data, how we are going to analyze this. You make your plan. And you really have to establish a systematic search strategy to select really the appropriate studies, and then you have to focus on outcomes for which you can extract data, because if you extract data of many different outcomes, you're not going to be able to pool them together. So you need to think about something that most of the studies have analyzed to be able to bring everything together. And you also need to think about study quality, how you're going to analyze it, which tool you are going to choose for that. And then, of course, you will gather this data, extract the data, analyze it. We'll have a look at that shortly. And in the end, of course, we want to write a report, an article to present your results, and if possible, to give recommendations for further clinical practice or further research. And then maybe you will be able to change the current practice of medicine because you found something completely new. That's actually the goal of it. Well, again, as I suggested, you need a strategy. You cannot just start and do it at random. You must have a plan. So you should write them in advance, but you must also know this is not like a randomized control trial where you control everything. You know you are going to measure this, to measure that, and you will analyze it this way. Here you are going to take the data that you will find in the literature. So maybe you will have to adjust your methods on the go because maybe you don't find the outcomes you want, or maybe the methods are not exactly the ones you expect. So you need a plan, but you also need some flexibility because you never know what you are going to find. And you really have to have a study question. You can actually use a PICOT format, just like we know, focus on patients, on the interventions, the control and the outcome, and then the time duration. You can have, it should have actually very specific questions. For example, in the recent work I did for this meta-analysis course, we focused on the catheter aortic valve implantation, comparing two approaches, transapical versus transfemoral, but only in studies where multivariate risk assessment models were used to adjust for baseline characteristics. So you can go very precise if you want to really assess a specific question. And it's important because if you are too broad, if you're like, let's have a look at that, you will end up nowhere. So you really need a question and links to it, criteria to include and exclude studies. You want studies that would like this, like that, and not this one, for example, based on choice of patients or the type of methods, the type of interventions, duration, whatever. You must think about it in advance, what you want to take, what you want to refuse. And this is the only way actually you can search the literature systematically, because you cannot just take one study because I like it and the next one I don't like it. No, I mean, you really need to have criteria. And for the data extraction also, you need to have a plan on how you are going to extract this data, which numbers are we going to take, what are we going to focus on, what is our main outcome. It's all important. And again, it's really important to focus on something that is measured everywhere, to have a common outcome that you can pool and then generate a pooled results that will be the main result of your meta-analysis. The quality assessment, I mentioned it before, you need to make a plan. What are you going to use? For example, for randomized controlled trials, you may want to use the Cochrane tool. We'll come back to it. And or again, you need a plan how you're going to analyze your data, how you are going to publish. And very importantly, you need a good team of researchers early on, because doing this alone is a hard work. If there are many, maybe there are thousands of studies to review, it's just terrible to do it alone. So have a good team of motivated people. You can share the work and share ideas and have a fantastic group work at the end. So, of course, you have to search the literature, as I told you. You have to be a bit systematic. There are many databases. The best known one is PubMed, but there are other ones like Embase, Corpus Web of Science, Cochrane, whatever, Signal also, and even more. You need to have a strategy first based on words. You want to search for keywords that are specific to your questions, to your patients, to your methods, to whatever you are looking for. Maybe you can exclude some keywords depending on your specific search. You will search this word in the title, in the abstract, the keywords of the paper, and importantly, don't forget the mesh. That's maybe something you have heard about. This is the medical subject headings. That's really important because actually, when a paper is archived, the librarian will put some keywords on it. It will say, well, this is a study about this and that, and it will put this into categories of articles based on broad topics, and you can use these topics to search articles. That's very powerful, because sometimes when you are searching for keywords, you may think of a word, but there is actually a synonym for it, and some papers may name the same thing differently. But if you use those broad categories instead of specific words, you have more chances of finding all relevant articles for your paper. That's important. And don't forget, you don't need to do it alone. There are librarians that can help you. They are happy to do that with you, to show you how to do it, to help you to refine your search. That's really a good opportunity to have a contact with librarians or maybe with other people who already did it. So don't do it alone if you never did it. And of course, if you do literature research, you have to have many people, as I told you, because if there are many papers to review, that's not possible alone. And don't forget to record each step, because everything must be reproducible. And in the end, you must be able to document how many papers you have found and analyzed and excluded and so on. So how do you do that? Actually, the flowchart that you see on the right, maybe I can move my face so that you can see it. Yes. Then you see actually how you do this, the process. You record, you find your references in databases, or maybe you also find references from reviews, because you have to look at recent reviews to find papers that you may not find in your systematic review, and you gather all those references. At this point, it's very useful to have a software just like EndNote or Mendeley, Zotero, whatever you like, to delete duplicates, to have one final list. There are also some softwares to help you for the systematic review, to share the work and so on, such as Covidence. That's also very nice. And then once you have your first list of papers, you just use a title and abstract to just do a first quick selection. Yes, no, that's quite easy. You can usually delete most of them. But then for a few last papers, you need to really download them, have a look at them, read the whole text and say, well, yes, this fulfills our inclusion criteria or not. And then in the end, you have your final list for your systematic review. And sometimes you cannot include everything into the meta-analysis. It depends on the data, but if possible, you can take everything with usable numbers into the quantitative analysis. Again, when you extract the data, you need to have a plan. You must prepare an extraction sheet, like an Excel sheet, very simply, like name of the study, and you want to have characteristics like number of patients, mean age, gender, whatever, depending on your questions and the outcomes, and some study characteristics. And don't forget to assess the quality of the studies. Again, you don't do it just like this. There are some scores. For example, the Cochrane tool for randomized controlled trials. You may have a look at, for example, blinding, randomization, or adjudication of events, and you have criteria on how to declare what is good, what is not so good, and in the end you can have a score or like plus minus for each study. That's important because then you can think about subgroup analysis. Maybe if you focus on good studies and exclude studies that have a lower quality, maybe the result is different. So that's something you need to analyze. And maybe if you see all the results are pretty bad, then whatever you will find in the end, you will say, well, but it's based on bad data, so we cannot really do much with it. That's an important point, actually. And of course, the funniest part is to do data synthesis, to generate those famous forest plots. You have one example, a recent example from my last study. But first, actually, I jumped to it, but I should first speak about the narrative synthesis. First, you should explain what you have found, and sometimes you cannot summarize it with numbers. And if possible, you do a quantitative summary of everything, and you try to produce pooled estimate of your outcomes. For example, if you have continuous variables, you may use the weighted mean difference. This is just the mean difference between groups, but with a weighting depending on the size of the study. If the units of measurements are different, you must standardize the results. That all looks very complicated, but don't worry, there are plenty of software that can do it for you, just like RevMap, which is free, or you can use Stata, you can use R, you can use whatever you like. And if you have binary outcome like events, death, infarction, stroke, or whatever, then you can assess relative risk, odds ratio, risk difference, hazard ratio, whatever you like, but you have to think about it in advance. What are you going to analyze? And then you can calculate it for each study. And that produces such beautiful forest plots when you have all those lines with squares. So each square is the result of a study. A bigger square means more weight to the study, so a bigger study. And the lines on the side are the confidence interval. So if you have a small study, you have big lines, so big whiskers, so large uncertainty, and in big studies, we have a big square and small bars because there is more certainty. And in the end, you will summarize all of this into the diamond, which you see on the lower part of the graphic. And then there is a global estimate of effect. In this case, this is a hazard ratio. with the confidence interval, with the p-value. You can even calculate the prediction interval, and you have also measurements of heterogeneity, but well, let's not go too deep there. An important point, you have to choose the model. Maybe you have heard of fixed effects versus random effects models. It depends if you consider that each study is exactly the same but made on different patients, or if each study not only has different patients but also different methods, different characteristics and so on, that generates a double level of heterogeneity. And actually in real life, in clinical science, you have both variations. You have different patients and different study protocols. So actually you should use a random effect models, which makes a broader confidence interval because it incorporates more uncertainty into the calculation to make it simple. And there are different models for that. Well, and you can also do other approaches depending on your question. Maybe you can do a dose response meta-analysis if you want to see, for example, the dose of a medication, of an exposure. If there is more effect with more exposure, you can also do this at the meta-analytic level. You can also meta-analyze diagnostic accuracy. You must do a specific calculation with sensitivity and specificity together. But it's another topic. It's also very interesting. Well, but I'm speaking too much as usual, so let's move on. There are also additional analysis that you can do, a lot of interesting things. You can measure heterogeneity. Maybe you have heard of Cochrane's Q and I-square that measures actually what is the amount of the difference between studies that are due to heterogeneity, and if this is too high, then in the end it's difficult to believe the result, because if there is too much difference between the studies, you need to have an explanation for that. One possible explanation is a publication bias, and here you have an example of a funnel plot, which is, as you can see, you have the dots, 1 dot per study on a funnel, and the dots that are higher on the graphic are big studies, and the dots that are lower on the graphic are small studies, and actually they should spread nicely around the middle line, particularly the small studies. And the phenomenon that we're looking for is if, for example, if all the small studies are on the same side. For example, small studies were published only if they had a positive results, and maybe not if they have a negative results. And you see that in an asymmetry of the plot, actually, and that would be a bias. And you could actually correct for it, for example, with trim and fill, but well, that's an important point. There are many tests for that, such as the figure test. You can also assess, for example, if each study had too much effect on the result with a leave-one-out analysis. You can do subgroup analysis in subtypes of studies. Maybe some of them had different impact, different results that could explain why some studies had completely different results. That's important. You can also do a temporal analysis. Maybe you will see that at which moment in time the results became significant, or if there are temporal trends. And also you can do meta-regression actually to see what is the impact of some study or patient characteristics on the results. Well, meta-regression sounds very funky, but actually it has limitations. So you always have to be very careful with that, because, for example, if you see that in studies with with older patients, there are more of the outcome, you don't know if actually the older patients were the one that got the outcome. This is kind of the ecological bias. It's the same problem as in ecological studies. So you have to be very, very careful with results from meta-regression. It's funky to do, but be careful with the results. And then in the end, of course, you have to report the results. You write a paper. Actually, writing a paper about a meta-analysis is just the same as for any other studies. You have the standard structure with the traditional methods, results, and discussion. Nothing very special. You present your methods that you have anyway planned in advance, so it's not a problem. It's very important, of course, to summarize your key finding and to evaluate how valid it is. Was the result significant? Did you have a problem with heterogeneity, a problem with study quality? Do you really believe what you find? Or do you think, well, we are not sure about it? Can you generalize it to all patients, or is it only valid for some subgroups and not for other groups? And in the end, what is very important with the meta-analysis, because we have some impact with it, is what should we do with those results? Should we do further research on the global topic or on specific subtopics? Or should we change our practice, and how? What should we do? That's really the points that you can really have an impact on the literature and practice. That's one of the very nice parts of meta-analysis, actually. And well, now you are ready to start. You are ready to join the Cochrane collaboration, or just start a meta-analysis and have fun and a lot of success with it. And if you have questions about it, I'm happy to help. As acknowledgement, as I wrote here, some aspects of this course were inspired by the course that I had by Professor Papatheodorou, which made a fantastic course about meta-analysis. So thank you and goodbye.